Artificial selection for trichomes in Wisconsin fast plants. The purpose of this lab is to observe how selection acts as a mechanism of evolution in Wisconsin fast plants, intentionally mate the plants with most trichomes to emulate directional selection. Hypothesis: if the hairiest plants from the parent generation are bred with one another, then the offspring generation will be, on average, hairier than parent generation, exhibiting a shift to the right. Both artificial selection and natural selection describe a change in the relative frequency of alleles. Artificial selection is a process by which humans modify species for desirable traits. Change occurs quickly and often causes fitness to decrease. Natural selection occurs in the environment and is devoid of human influence. Change tends to be slow. Selected traits improve chances of survival and reproduction. Essentially, circumstances determine the advantageousness of various traits, resulting in the survival of the fittest. In population genetics, natural selection, not artificial selection, is characterized by directional, disruptive, and stabilizing selection. Artificial selection may emulate any of the aforementioned selection types. Stabilizing selection entails the removal of extremes from the population. The frequency of intermediate individuals increases. Disruptive selection favors outliers. Intermediate organisms are at a disadvantage. Directional selection shifts the population makeup to either extreme of the distribution. Dr. Paul H. Williams developed rapid cycling Brassica rapa, more commonly dubbed Wisconsin fast plants, through 20 years of genetic research. Dr. Williams' goal was to increase the resistance of plants from the family Cruciferae to diseases such as clubroot. To obtain results more efficiently, he bred Brassica rapa with 6 similar Cruciferae species. The offspring had, on average, 28-day life cycles. These plants were then refined into the Wisconsin fast plants used in classrooms and laboratories today. Fast plants can express over 150 genetically modified traits. Materials: 30 parent generation fast plant seeds, 40 fertilizer pellets, 20 used per generation, flower pot, potting soil, water source, bamboo skewers for labeling and support, graduated cylinder for transferring water, plant growth tent with screen and light source, plastic tub, magnifying glass, pencil and paper for records, paintbrush for pollination, ruler, and diluted isopropyl alcohol. Time spent on this experiment may vary. Since Wisconsin fast plants take around 28 days to complete their life cycle, offering at least 2 months is ideal. Note that not all materials will be used on the first day. 1. Fill a flower pot 2/3 of the way with damp potting soil. 2. Evenly distribute 20 pellets of fertilizer onto the soil. Cover the fertilizer with a 1 cm thick layer of damp potting soil. 3. Count the number of fast plant seeds given. Record the number. 4. Evenly distribute the seeds onto the potting soil. Cover the seeds with another thin layer. 5. Label the pot with necessary identification. Place the pot into the plant growth tent that should already contain a light source and the plastic tub 1/3 full with water. The plant growth tent screen will reduce the incidence of unintentional cross-pollination. 6. Assign one person from the group to ensure that the plant does not become dehydrated. That person should add water to the tub whenever necessary. 7. Wait for at least 1/3 of the seeds to sprout. Then take the fast plant out of the growth tent. 8. As a class, decide the locus for counting plant hairs. Example: 1 cm from the main stem, from the first leaf, or from the tallest leaf. Any zone that is replete with trichomes may be chosen. 9. Use bamboo skewers to number the plants and provide support for the tallest ones if necessary. 10. Count the number of trichomes in the experimental loci. Using a magnifying glass will result in a more accurate count of the hairs. Multiply the amount of hairs by 1 cm divided by the experimental locus length. 11. Take pictures of the fast plant and its hairy loci. 12. Evaluate class data. Apply pollen with a paintbrush from the hairiest plants to your own fast plant. This cross-pollination will result in artificial selection. Rinsing the paintbrush with alcohol before and after brushing from anther to stigma will result in more accurate data for the offspring generation. 13. Wait until pods form and dry. Remove the seeds from the pods and throw away the parent generation plants, soil, and fertilizer. 14. Repeat steps 1 through 11. Keep the same flower pot. No new label is required. Use the same loci for counting. 15. Throw away the plants and return materials to where they belong. Clean up the work area.
As the plant numbers are arbitrary, there are 2 ways to evaluate the data. On the graph to the right, consider the sets for January 17 and April 7 as distinct. A comparison of the data suggests that offspring generation is significantly hairier than the parent generation, matching the hypothesis. 2 plants, labeled 6 and 7 from the parent generation, grew exceptionally well, having 35 hairs per centimeter each. But half of the parent generation plants, labeled 1, 3, 5, 8, 13, 14, 15, and 16, had 0 hairs. The shift in the average number of hairs per centimeter to the right was 6.19 to 23.44 for our group and 12.92 to 20.59 for the class. The percent increases are 378.7 and 160 respectively. The second way to evaluate the data is to plot frequency, or the number of plants, against hairs per centimeter. 0 hairs per centimeter plants were very common for all groups. If plants were accounted for individually, the 0 to 5 range would have easily triple or quadruple the frequency of the 6 to 10 or 11 to 15 ranges. As such, evaluating each group's average as a single unit frequency will result in more representative data and a better bell curve line. While a shift to the right exists, directional selection is not perfectly emulated. Points do not fall into the bell curves due to few notable outliers in class data. The curve line for the parent generation is understandable, since no artificial selection took place when raising the parent fast plants. The amount of trichomes per centimeter varies significantly. Plants with approximately 6 hairs, for instance, appear as frequently as plants near the 12.92 average. The high frequency of offspring plants in the 21 to 25 hairs per centimeter range may be attributed to cross-pollination. All groups took pollen from the same plant, the one with the most hairs. The outliers are the offspring hair counts for group 7 and 8. Group 7 experienced a decrease in hairs from 19.17 to 13, whereas Group 8 experienced a drop from 13.09 to 11. The most likely explanation for this trend is that Group 7 and Group 8 use seeds from a plant with relatively few hairs. Eliminating these outliers can be done with a simple fix mentioned later in the errors and improvements. Conclusion. In this experiment, our group observed how selection acts as a mechanism of evolution in Wisconsin fast plants. We mated our plants with the hairiest ones to emulate directional selection. We hypothesized that if the hairiest plants from the parent generation are bred with one another, then the offspring generation will be, on average, hairier than the parent generation, exhibiting a shift to the right. Choosing hairier plants when breeding is very much like applying a selective pressure. Outside of the laboratory environment, people do not care much about the presence of trichomes in flowering plants. Breeders instead select for flower color, shape, and size. The natural world, however, does favor trichomes. Trichomes exhibit toxic and deterrent effects. They thus play an imperative role in plant defense against pests. An increase in trichome density is detrimental to feeding, larval nutrition, and ovipositional behavior of insects. Furthermore, trichomes interfere with arthropod movement across the plant surface, reducing herbivore access to leaf and stem epidermis. Trichomes are evolutionarily diverse. They can be straight, hooked, spiral, branched, unbranched, glandular, or non-glandular. Glandular trichomes secrete flavonoids, terpenoids, and alkaloids, secondary metabolites that poison, repel, and trap insects, providing considerable chemical and structural defense. The density of trichomes in specific loci of plants does not change during growth. To survive an insect infestation, plants may develop more leaves, inducing trichome density increases between 25 and 100%. Adaptive trichome density changes occur within days or weeks after herbivory. That said, plants obtaining higher densities of trichomes before herbivory are more likely to reach reproductive age, creating directional selection towards the high trichome densities. If a greater sample size were taken, and if the Wisconsin fast plants were exposed to insects over more generations, artificial selection would better exhibit directional selection. Our removal of the plants with the fewest hairs from the breeding population helped illustrate Darwinism's survival of the fittest. Organisms with more favorable traits more often reach reproductive age and pass on successive genes to their offspring. Indeed, the roles nature and time play into the development of directional selection curves should not be overlooked, for data is far from the expected values, only verifying the hypothesis that a shift to the right exists. Errors and improvements. The first change that could be made to experimental design is counting trichomes after the fast plants reach reproductive age. Step 10 involved multiplying hairs counted by a factor of 1 centimeter by locus length. If a sprout had 5 trichomes in a locus an eighth of a centimeter in length, the recorded trichome number would be 40, unlikely reflecting trichome density of the plant as an adult. Simply waiting for more plants to grow a centimeter of locus will result in fewer outliers. As with most experiments, increasing sample size should bring the data closer to expected points. If all biology classes kept the fast plants in the same growth conditions and amalgamated data, the frequency versus hairs per centimeter graph will get more data points, eliminating the need to section the x-axis off into ranges, in turn producing more valid curves. One possible human error is miscounting the number of trichomes. The hairs were indisputably difficult to see, even even through the magnifying glass. However, skipping and double counting hairs could not have skewed the data too significantly. Slight overestimates and slight underestimates statistically balance out. Still, encouraging more accurate hair counts would not hurt. Adding dyes could make the hairs more visible. Tweezing out counted trichomes could eliminate double counting. None of these actions should affect the genetic makeup of the plant. To implement these revisions, however, the experiment would demand more time and materials.